We've talked about some of the important proteins that are involved in transport of molecules across a membrane. So let's talk about how these molecules actually move. So there's a couple ways that they can move. One is through a process through diffusion. So in diffusion, a molecule moves from an area where it is highly concentrated towards an area to where it is less concentrated. So I like to think of this as individuals leaving a stadium, or think Busch Stadium or Enterprise Center, and there's a high concentrations of individuals there, and by the end of the game, people just want out, and so they spread out into the city where they are in a lower concentration. So diffusion works the same way. So here we have an example of a food coloring, a dye, being dropped into a glass of water. Now the initial drop is an area of high dye concentration. So there's a lot of dye molecules in there. And over time, if you watch it, those dye molecules will spread out. Now you don't have to do anything. They actually are moving. All those little molecules are moving within the water, and they're bumping into each other and causing the molecules to spread out into the water. So here we've got high dye concentration. They bump into each other randomly and slowly spread out. And you've probably observed this yourself. Now they move until they are evenly distributed. So basically that means they move from an area of high concentration to an area of low concentration until they are in equilibrium, which just means that everybody's spread out easily and they're all moving sort of randomly back and forth. So this happens across a membrane as well. So there are two types of what we call passive transport. So passive transport means there's no energy involved. Molecules simply move based on their concentration. So we just talked about simple diffusion. So over here we have oxygen molecules. So these oxygen molecules are in high concentration here. These oxygen molecules are actually really small and non-polar, which means they can move easily, excuse me, through the plasma membrane, and they will move from where they are highly concentrated to a region where they are less concentrated. Now the other side is carbon dioxide. So the cell needs to bring in oxygen, but it needs to push out carbon dioxide. So carbon dioxide will move the opposite way against its chemical gradient. So carbon dioxide is also small and nonpolar, so it's going to move freely across the membrane from an area of high concentration to an area of low concentration. Now we can also have something called facilitated diffusion. Facilitated diffusion just means that it's diffusion happening through a mediator or through a channel. There's in this case, a protein that's embedded in our plasma membrane, and those molecules that need to come in are highly concentrated outside and less concentrated inside. And so they'll flow through this channel protein from their area of high concentration to their area of low concentration. Now, this can also go both ways depending on the protein. There are different types of proteins. There are these channels where molecules can just sort of flow freely, And we also have molecules called carrier proteins. And the carrier proteins actually change shape in response to the molecule. So they're a little bit more specific. But either way, the molecules are moving from high concentration to low concentration. So the difference between simple diffusion and facilitated diffusion is that in simple diffusion, molecules can just move across the membrane directly, but in facilitated diffusion, they need some sort of channel or carrier molecule through which they can flow. And the reason for that is that they are usually large and charged or polar, and so they would not be able to move directly through the plasma membrane. We can also have active transport. So active transport is transport that requires energy. So the energy molecule in the cell is a molecule called ATP, or adenosine triphosphate. And the ATP molecule has the ability to supply chemical energy to a variety of chemical reactions within the cell. So primary active transport, one of our types of active transport, is where ATP is used directly by the molecules involved as an energy source for a particular process. So in this picture here, we have our primary active transport. So ATP binds to this protein, this transmembrane protein, and in doing so allows that molecule to pass allows that protein to move molecules. Now, the reason that we need energy in this case is that we want to move these molecules against their concentration gradient. So remember that diffusion tells us that molecules are going to move from high to low concentration. If we want to move the other way, we need energy to do that. It's sort of like if you're trying to shove more things into your suitcase. You have to put a lot of energy in to get more in because there's already a lot of clothes concentrated in that suitcase. So it's the same way with a cell. Sometimes it needs a higher concentration of molecules in a particular location, and so energy is required to build up that concentration gradient. We can also have something called secondary active transport. In secondary active transport, molecules are moved using energy indirectly. So in this case, we have a molecule, two molecules moving. One is this blue molecule here, and this blue molecule is moving along its concentration gradient. So the power here for moving the other molecule, this yellow molecule, actually comes from the movement of the first. So as the diffusion gradient causes the blue molecule to move, it actually provides energy to store up this yellow molecule against its concentration gradient. So we have these channel proteins and these carrier proteins that allow molecules to move through the cell. They are sometimes specific, sometimes not, but they are always moving single type of molecule. So maybe it's a large polar molecule, or maybe it's a specific type of ion or something like that. We can also sort of bulk transport particles into the cell. Now, there are two different processes. One is called endocytosis, and that's the bringing of particles in. The other is exocytosis, which is the process of removing large particles from the cell. So endocytosis is basically the cell engulfing or enclosing around a large particle. So the plasma membrane will actually extend around the particle it's trying to bring in and then bring it in. This is how we got that endosymbiosis theory. The plasma membrane sort of extends around something and brings it in. So there are a couple of types of endocytosis, and we can think of these as cell eating and cell drinking. So phagocytosis is what I like to think of as cell eating. So on the left-hand side there, we have a paramecium, which is a large single-celled eukaryotic organism. I'm sorry, what did I say? It's an amoeba. I may have said something incorrectly. That's an amoeba eating a paramecium, which is another type of cell, another eukaryotic cell. And you can see that it is extending its quote-unquote arms around this paramecium, and eventually it's going to bring it in and basically fuse it into its cell. So phagocytosis is bringing in of particles. So over here we have the particle, we have the plasma membrane sort of pulling in and pinching around the food particle that it's trying to bring in. And eventually it pinches off sort of a little bud inside the cell called a vesicle. Now a vesicle is just a little transport pocket of cell membrane that can move throughout the cell. So similar to phagocytosis is the process of pinocytosis. Pinocytosis is the same process except that it is moving in of fluids. So you still get the plasma membrane pulling around whatever particles you're trying to bring in and producing a vesicle, but this time with dissolved nutrients from the fluids around. We can also have something called receptor mediated endocytosis. So this is where you have sort of a little traveling packet. In this case, this is a vesicle filled with cholesterol molecules, and you'll notice that there are these little identifier fingerprint proteins on the outside. Now this allows the cell to recognize those cholesterol particles, or packets, if you will, and specifically bind to the membrane of a cell that needs them. So receptor-mediated endocytosis is the process where you use receptor molecules to identify what you want to bring in the cell. So there are a couple different types of proteins. We've got the low-density lipoproteins and the high-density lipoproteins. And remember, cholesterol is really important in regulating fluidity of the plasma membrane. This receptor-mediated endocytosis is the process by which the little particle containing your cholesterol molecules can actually be brought in to the cell. So cholesterol is then used for the membrane. It can also be used elsewhere in producing hormones. So cholesterol is actually a precursor to our sex hormones, estrogen and testosterone. So there's a lot of uses for cholesterol in the cell. There are different types of receptor molecules. So one receptor might be for insulin, another for cholesterol, but they are the point of them is to make sure that the cell brings in the molecules that they need. So these LDL receptors are really important in something called hypocholesterolemia. So this is when individuals have high cholesterol. They have this disorder, and there is actually a genetic type. So this is called familial hypocholesterolemia. And this disease is actually a lack of these receptor proteins. So individuals that lack these receptor proteins actually end up with a sort of a blockage of cholesterol. So this can lead to cardiovascular disease or heart disease. And this circulating cholesterol sort of builds up in arteries and impedes blood flow. So just like we saw with cystic fibrosis and faulty membranes, we also have membrane problems in this disease as well. We don't have the correct receptors to properly bring in and process cholesterol, so to get the cholesterol out of the bloodstream. So cholesterol is really important, but it can be a problem when your body can't utilize it correctly. Of course, symptoms of heart disease can lead to reduced blood flow to the heart. It can lead to reduced blood flow to the brain, things like that, causing a lot of problems for the individual. So we talked about bringing things into the cell. Exocytosis is the process of removing things from the cell. So basically, it's a way to get rid of waste. It's a way to get rid of cellular products that are needed somewhere else. So let's say we have some cellular material that needs to be broken down and shipped out of the cell, or we have some proteins that have been made and they need to be packaged for use elsewhere. So just like in endocytosis, we're going to utilize something called a vesicle, which is basically a little mini plasma membrane packet with whatever we want to move out of the cell. So that little vesicle encloses the particle that needs to be moved. And then in the reverse process of endocytosis, it merges with the plasma membrane and releases it on the other side. So up here, we have an image from the pancreas. So the pancreas contains hormones, and it actually produces insulin. So the pancreas can release insulin into the bloodstream via this process of exocytosis.